So I'm going to be talking today about what I say Bryophilous ascomycetes, a microscopic El Dorado. So microscopic because really, if you want to study these things, you need to use a microscope. And El Dorado because there's just so many of these fungi, both in terms of the species, but also the different ways that they interact with different bryophytes. It's really unknown, a sort of gold mine of amazing biology just waiting to be found. So I hope I can entice you with this sort of whistle-stop tour of these species. You can find me on Twitter if you like, and I'm based at the University of Bristol. I'm a PhD student, and I'm doing a PhD in plant development, and this sort of studies on bryophyte parasites is my sort of hobby. I do it in my spare time. So I'm not an expert on this stuff necessarily. I don't, it's not my job, but I have been involved in it now for a number of years, and really keen to encourage other people to get interested as well, because it's just a seriously interesting field. So the structure of today's talk is I'm going to outline Bryophilous ascomycetes a bit later on, but I'll first talk about myself and like how I got interested in this really obscure field of biology. I'll give a bit of basics on bryophytes, the plants I look at these fungi on. I'll then introduce the fungi themselves, this group of fungi, the ascomycetes on bryophytes. So these aren't things like mushrooms and toadstools, which the vast majority of people will be familiar with. It's little what are called sac fungi that produce their spores in a different way to mushrooms. They're smaller, they're sort of a bit more blobby. And I'll talk, again, how I study them microscopically and also in the field. I'll talk about fungi on liverworts, fungi on mosses, and I'll conclude with a few things we can learn from studying these fungi that are more than just identifying species. So to get started, how the heck did I get interested in this random field of tiny fungi on bryophytes, on tiny little plants that, barely anyone knows about the plants. Most people probably didn't know that these plants can get infected with fungi. Well, it probably starts with the fact that I am originally from South Africa. So my family is British, but I was born in Johannesburg, and I grew up there for 19 years in a very different place to South England, where I moved to the Isle of Wight, to be specific. So in Johannesburg, we had dry, barren winters where everything just turned yellow and died, and it was just cold and it never rained. And in summer, we'd have these really hot days, and then at sort of 5 o'clock every day, we'd have a massive thunderstorm with, thunder, lightning. It would chuck it down with rain for 15 minutes, absolutely flooding the place. And that would be the only rain really that we get every year. And obviously, the UK is a bit different to that. It's green pretty much all year round. It never stops raining, really. And it's a lot, it's just very different. What you don't see in Johannesburg are plants growing on other plants, growing on rocks, just sort of, on the soil. Just everything was so green, and there was all this creepy, squishy stuff that I'd never really noticed before. And it was, just such a different place compared to where I grew up. And one day while I was studying at the University of Portsmouth, and I was living on the Isle of Wight, I came back home one day, and I had the choice of either getting a bus home or spending my bus money on chocolate and walking home along a 4 or 5 mile cycle track, which is this picture here on the right. And obviously I did the very good decision, which was to, eat a chocolate bar and get some exercise. And being fairly new to the UK, I think it was my very first winter, it was December 2015. I was walking home along this track, and obviously it started raining quite hard, actually. And I didn't have an umbrella or a raincoat. So I took shelter under one of these Victorian arch bridges, because this cycle track used to be a railway line, and there are all these old bridges along it. And I noticed along the wall of one of these bridges a really interesting plant, this liverwort, Connocephalum conicum. And really, to me, who'd never seen anything like this before, it looked like a seaweed was growing out of the brickwork. The bridge had seepages on it, and there were sort of all these this sort of plant stuff covering it, this green stuff. And it was just, I'd never seen anything like it before. I'd gotten a microscope for my studies in biology at the time, and I took a little piece home and looked at it under the microscope and saw these big plant cells and air pores and all sorts of stuff. And I thought, wow, this is such a weird plant. And I crushed it, and you can smell this really vivid smell that this plant produces. And I was just captivated. I'm not sure why, but everyone has a funny origin story when it comes to natural history, and well, that's mine. I got interested in not just bryophytes. I explored a few different avenues of what's called cryptogamic botany, so the study of things, sort of ignored organisms such as fungi, lichens, bryophytes, algae. And this was really because of two people, but mainly because of Colin Pope, who's in the picture here, who's an excellent naturalist based on the Isle of Wight. And he really helped me learn my mosses, or learn how to identify bryophytes. He, showed me records, how to look things up in identification guides, habitats, all kinds of things. And if it wasn't for Colin's help when I was getting started, there's absolutely no way I'd have been able to get anywhere with any of this stuff. It's really not easy to get your feet off the ground, especially if you have no background in botany whatsoever. I should say, as thanks to Colin, I named a species after him of fungus. So hopefully we can I can thank him for what he's done, which was very kind of him. But Colin is not only an excellent naturalist in say flowering plants, for example, but he actually wrote a lichen flora of the Isle of Wight. So while mosses and liverworts are proper land plants, lichens are a symbiotic association between a fungus and an alga or a cyanobacteria, and they produce these composite organisms that are often really vividly colorful and grow on trees and twigs and rocks, all kinds of things. And many people who are interested in natural history will be familiar with lichens, and they're just amazing organisms. And what I found really fascinating was that Colin told me that not just where, you've got a fungus and an alga interacting together to form this really strange thing, but they get parasitized by fungal parasites as well. So other fungi come on and start killing these lichens and eating them. And here's one that turns the lichen sort of reddish color. And there's actually one on this picture as well, which turns the fruiting structures of the lichen black. And I just thought, wow, there's fungi killing these fungi on algae and all kinds of things. And I was just sort of awestruck by these interactions. And I thought to myself, what about bryophytes? Do they have any fungal parasites? And all the kind of textbooks and things that I'd read at that point suggested that they didn't, and that they, like that liverwort that I mentioned earlier, they have all these compounds in them that are antiseptic, and they must be amazing at producing, maybe the next generation of pharmaceutical drugs, because nothing eats them and nothing kills them. Until John Norton, who's an excellent botanist and bryologist, picked up this moss with a fungus on it. And I thought, wow, so there are fungi on mosses. And it took a while to identify this fungus. And I wasn't very good at microscopy or mycology at this point. I had no previous experience in sort of mushrooms or anything like that. So it was quite a sort of uphill battle learning about these things. And really, for the Bryophilous fungi, this got me into literature by Peter Derbler, who's the world expert in Bryophilous ascomycetes, which is the group of fungi I'm interested in. And it was really through reading his papers, and his research, which is just meticulous, amazing, and spans, a huge career of just great research, that I was able to persist and get interested in these things. And really, Peter's correspondence and his help has really been, not just his papers, has been really fundamental for me learning about these things. So really, for me, I'm a bryologist first and foremost, and I have a sort of what I'd call an interest in holistic bryology. So I'm interested in basically how bryophytes work and how they interact, broadly speaking. So how they work comes down to their genetics, development, reproduction, physiology, and this feeds into their evolution, which is something I'm very interested in as well, how plants evolved on land. And also how do bryophytes interact also links with their evolution, but this is more to do with their ecology. So bryophytes are very specific in the habitats that they grow in and the things that they grow on. So many bryophytes only grow on trees, or they only grow on a certain type of soil in a certain type of environment. And this is really interesting stuff. But really, the focus of today's talk and the sort of research I do on the side of my PhD, which mainly focuses on the genetics, development and reproduction and evolution, is this side here on symbiosis. So how do bryophytes interact, and how do they interact with fungi? How has that shaped their evolution, are some of the sort of questions I'd like to maybe investigate in the future. And also how do their interactions with fungi influence their development as well? So I'll be talking a little bit about possible avenues and looking at that a bit later. But firstly, I need to tell you, what are bryophytes? So bryophytes are land plants. They are plants as sure as a flower is. They comprise of three groups: the mosses, the liverworts, and the hornworts. I won't really be talking much about hornworts today because there are only 4 species in Britain and not many in Europe. And there aren't a huge number of ascomycetes that grow on them. And they're quite rare, so I don't have a huge amount of experience with them. Bryophytes lack flowers, they lack seeds, they lack xylem and phloem, which are the transport mechanisms for water and food that vascular plants have. And they lack true roots as well. So this makes them quite small usually and quite delicate. They're ground hugging, they require water for reproduction. They have very different methods of dispersal and reproduction compared to vascular plants. But they are plants. So many people say, what's a bryophyte? Is it an alga? Is it a lichen? Is it a fungus? No, it's a proper land plant. They've just adapted to living on land differently to the vascular plants, so differently to grasses, flowers, trees, ferns, lycophytes. And they diverged from this lineage of vascular plants and evolved separately and have been doing so for 450 million years. So bryophytes have adapted to living on land in different ways to the vascular plants. A lot of people would say bryophytes are lower plants, but really they're not. They're just different. They might be lower because they're smaller most of the time. And bryophytes really have 2 main ways of living on land. While vascular plants are generally desiccation resistant, so things like trees can survive even when the air is not very humid and it's windy or whatever, bryophytes will shrivel up usually. So bryophytes can, you'll know if you've ever studied mosses at any point, you'll have heard that mosses have a desiccation tolerance, so they can dry out. And you can see here a moss on some stonework or in a pavement, and it's dried out completely, and it goes into suspended animation. And if you were to add water, it would come back, go green, and photosynthesize. The other strategy bryophytes use is desiccation avoidance. So these species will either grow in places that are permanently damp, like riverbanks or something, or they will have life cycles where they're only growing in very damp times of year. So unlike flowers, for example, which often come out in the spring, flower and die at the end of the summer or whatever, a lot of bryophytes will come up in the autumn or in the winter and die over the summer period and not be around. So it's kind of an inverse life cycle. So a lot of botanists get interested in bryology because there's nothing to do in flowering plants in the winter, but all the bryophytes come out. And bryophytes are hugely diverse morphologically, and they have a huge amount of adaptations to living on land. So here's just a snapshot of some bryophytes. So here's a hornwort. There are a few mosses here, so a red moss, some other mosses on the side here, really weird looking moss, one that looks a bit like a flower. Here's a liverwort that's delicate, grows on rotting wood. And here's another liverwort which grows up in mountains on exposed rocks. You can see a huge amount of variation and complexity in these plants. And I'm not really going to go into the nitty-gritty and the differences between mosses and liverworts here, but really you can think of liverworts as being flat plants that are sort of more two-dimensional. It's not really accurate, but for the purposes of this talk, you can see here, this thing all has its leaves going out in one row, whereas mosses are more three-dimensional. So you can see there are leaves around a central stem. And this feature makes liverworts a bit easier to study or screen for fungal infection than mosses, because mosses are, you have to dissect more. I'll just take a sip of water. Right. So some generalized bryophyte anatomy, really. So this is a common moss, Tortula muralis, that you'll see on walls all over the place. In Britain, and even actually in South Africa, you can find this moss. It's super common. And really, if you get down to the level of a moss, they look very much like a normal plant. They have root-like structures called rhizoids, which play a role in anchoring the plant to whatever it's growing on. These can absorb nutrients, and they can also interact with microorganisms in, say, soil. They have stems. They have leaf-like structures called phyllids, which are generally 1 cell thick in many species. So here's a moss leaf. They're wonderful to look at under the microscope. They have reproductive organs, and I'm not going to go into it, but their reproduction is quite different to flowering plants. They produce egg cells and sperm cells, and the sperm cells have flagella and they move around in water. So mosses need water for reproduction. And fertilization of the egg by the sperm cells makes these sporangia, which you can see in the picture here. And the purpose of the sporangium is to make spores, and these spores can blow away in the wind and make a new plant on another brick or wall somewhere. And that's the main way that many mosses reproduce. And microbes, specifically fungi, have evolved to interact with all parts of bryophytes. So you can find, for example, I work on fungi that produce fruiting structures, so basically blobs that contain spores. But all sorts of microbes interact with all different parts of bryophytes. So here's a couple that interact with the sort of rooting system. This one interacts with the stalk of the sporangium. This one goes inside the sporangium and infects the developing spores. This one's on the leaves, and this one exploits the male sex organs. So it's a parasite that specifically pretty much kills male mosses. Really, 99% of the described species that we know that are specific to bryophytes, so obligate bryophyte sort of interacting species, are fungi. There are about 500 known taxa and thousands of undescribed species. Now, I say that with, you know, I don't know how many there really are, but there are about 20,000 different bryophytes in the world and only 500 known species. And considering that several liverworts, for example, can host up to 10, 15 different host-specific fungi, it's likely that the number of Bryophilous fungi is super high, and we've barely scratched the surface. Another thing that's really good about studying Bryophilous fungi compared to any other fungi that people study is that you can observe the mycelium of the fungus. So as many mycologists will know, if you go into the forest and you find a mushroom, the mushroom is just the fruiting structure or reproductive part of the fungus. The main part of the fungus is the mycelium, which lives underground or in tree roots or whatever in the soil, and is microscopic, invisible and difficult to see and study. Sorry, just got a bit of a cough because I've been preparing this talk all day, and now it's catching up with my throat. So here is a fungus living inside liverwort cells, and you can see it invading adjacent cells very clearly. Here is another fungus living on top of moss cells kind of randomly on top of them. And here is another fungus which is able to thread itself between the grooves of cells. So it's still on top of them, but it's going between the cells. The anticlines of these cells is what this area is called. Here's another one that I've stained that grows on the outside and also infiltrates the inside and forms a branched assimilatory fungal structure to interact with that cell. This fungus is cool because it keeps the cell live and uses probably some sort of molecular mechanism to exploit that cell, get that cell to give it nutrients, but not killing it, whereas this one kills the cells. I should say that these three images were taken by just taking a liverwort leaf, putting it in water, and examining that leaf under a high power objective on a compound microscope. No staining, no sectioning or any crazy preparation was required for this. Because the leaves of liverworts are one cell thick most of the time, it's very easy to see this stuff without having to do anything crazy in the preparation of the material. And these fungi, the mycelium is naturally brown, which helps. This one, the mycelium is transparent, so I've stained it in a stain called cotton blue, which allows you to visualize the fungal structure. So the stain gets into the fungus and turns it blue. So it's really nice. But so that's one thing I think studying Bryophilous fungi, you can't really say in many other groups of fungi. You don't study the vegetative mycelium very often as a mushroom biologist, for example. So you get to explore an area of mycology that's kind of very different to anything if as a mycologist you may have explored before. So yeah, as I alluded to, I deal with ascomycetes, so not actually anything that makes a mushroom or a toadstool. So you can remove mushrooms, bracket fungi, and rusts or whatever from your mind. You can think more about Bryophilous ascomycetes as blobs of various sizes. So to illustrate the diversity of these things, I study mainly 6 classes of ascomycetes. There are a lot more and their taxonomy might change, but mainly it's things like pezizomycetes. So if you're a mycologist, you might know Pezisa, these sort of cup-shaped fungi. These are some of the biggest and most conspicuous Bryophilous fungi. We also have Leoshiomycetes, which are related to things like the ash dieback pathogen, Hymenoscyphus fraxineus. This is a really common and increasing problem in Europe now, where a lot of our ash trees are really suffering from this parasite. We have some lichen fungi. We also have Dithidiomycetes. So mycologists may be familiar with these smiling sort of black lips that you see on woody plants sometimes, like Hysterium. We have Sordariomycetes, things like Nectria, and Uroshiomycetes, which are really interesting Bryophilous fungi, I think. And they're related to things like the penicillium, the mold that actually produces penicillin. So really, a lot of different ascomycete groups have evolved to interact with bryophytes independently. There's not just one group of fungi that does it, lots of different fungi do. And even within these groups, there have been repeated bouts of evolution where fungi have gone and jumped onto bryophytes and managed to interact with them and radiate into interesting new fungal groups. So I'm going to briefly talk about how I study the morphology of these things. I'm not going to go into a huge amount of detail. If you want to find out more, you can read this paper that I wrote in Field Bryology, which should be available online. And any paper by Peter Döbler, his methods are basically this. So you need infected bryophyte material that you want to look at for fungi. There are 2 ways of getting this. The first is field work. You get fresh material. You can either go into a forest and randomly collect some bryophytes and see what you find. You can do a targeted approach of saying, I know this fungus grows on this moss, and I'm going to go collect this moss until I find the fungus. You can preliminarily screen for fungi if you know what they look like using a hand lens. Some will be visible, some won't. And you can also look for symptoms of infection. So many fungi won't cause symptoms, but some do kill the moss, and you can see big patches of death in moss and liverwort populations in the field quite clearly. The other way of getting material is through herbaria. And obviously, for someone like me, I keep a lot of my specimens, so that eventually becomes herbarium material. There are advantages and disadvantages of things being fresh or dead. So many structures will deteriorate when things die, and it won't be quite as good for some forms of, or some characters of the fungi won't be as good in dead material as in fresh material. But there are many advantages to looking at herbarium material, specifically being able to assess biogeographical trends in these fungi. So you can screen hosts that have been collected, say 100 specimens of a moss from all across the world, and look at that moss and really find out all the different fungi that grow on it that you can find, which is a really powerful tool, because you can say how frequent fungal infections are on particular mosses, maybe where some species occur in particular areas and not in others. So it's a really powerful tool for examining something. And I think an integrated approach of finding fresh material, getting a good description of something, and then going into the herbarium and looking for more of it in collections is a really good way of learning a lot about these fungi. It's not suitable for everything. Some fungi really don't keep well, but for those that do, it's a really powerful way of studying these things, and really quite exciting, because the vast majority of moss herbarium specimens have never been looked at for fungi. They are in the herbarium, they're sitting there with fungi on them, and no one's ever looked, especially not the bryologists who collected the material. Once you've got host material, you should have a look at it closely under a dissecting microscope. You should hydrate the material, you can remove debris, you can put the moss in a little sieve and run it under a tap to get rid of any dirt, pollen, invertebrates, or algae. So often you get a lot of debris on mosses that can look like fungal fruiting bodies that aren't. I should say these fungal fruiting bodies are often in the region of about 100 to 250 micrometers wide. So very small most of the time. There you can give or take, some go up to several centimeters in exceptional cases, but most of the time the ascomycetes, especially the specialists, are very, very small. So you need high power, relatively high power even in the dissecting scope to see them clearly. And sometimes you see algae and you can't even tell whether it's a fungus, so you have to sort of dissect it, examine it under high power to see that. When you have got moss material that you're kind of looking through under the dissecting scope for fungi, you'll need to kind of move it around, use forceps, dissecting needles, whatever, to really look through the plant material to find fruit bodies. You can examine it under high power, which you need to do if you want to see fungal spores or any of the structures that are actually of taxonomic value. And you need to use oil immersion and 1,000 times magnification. This is standard on most compound microscopes. So anyone who's a mycologist already who does mushrooms, for example, will probably be using oil immersion for mushroom spores. So maybe for bryologists, any bryologists in the audience who don't use oil immersion that frequently, that's probably more of a step up in microscopy. I should say examine everything in water first, do your measurements, and then if you need to apply any stains or reagents or chemical tests, you should do that afterwards. And really, with these fungi, it's good to make drawings, document what you found, keep things in a herbarium, send them to me if you want, if you're in Britain, because these things really aren't known about very well. So any collections, even of things that are relatively common, are valuable and can really help us to improve our knowledge of these things. So I hope that gives you a little snapshot on how I study these things. If you want to find out more, yeah, look at these, look at any of the resources. I should say as well that the fruit bodies of many of these fungi are tiny, as I mentioned before, often maybe 50 to 250 micrometers wide. You can't see them with the naked eye most of the time. And also, they're delicate structures and they're often hidden on the moss or liverwort that they're on. They're not always just going to be in plain sight as soon as you look under the microscope. So it's important to know what you're looking for, the size and the color of the fungal fruit bodies, what host preferences the fungus has. You know, you're not going to find specific fungi for one liverwort on a completely different moss. So you need to learn a bit about the bryophytes. Now, if you're a mycologist, don't be freaked out by this. There are a lot of very common bryophytes that are super easy to identify that are absolutely infested with fungi. So wouldn't worry there. And you need to know, as I alluded to, where on the plant to look for fungi. Where do they develop on the plant? So for example, here is a long-lived moss, Hylochonium splendens, and basically it grows in woodland or in heathland. And the lower regions of the moss here, these sort of brownish regions, you can kind of see the annual growth of the moss where it's green for 1 year, yellow next year, another year, and another year. So perhaps 4 years old this moss might be. And it's this sort of region where it gets darker where you might find fungi or fungal fruit bodies anyway. There's no use of just picking up this green bit, having a look at it and saying, oh, there's nothing there. This fungus doesn't have anything. So really, if you want to look for these things, you have to know where on the plant to look. If you want to find out more, I strongly suggest this paper by Peter Döbler. It's a really good paper. I'd say it's probably the one that got me really stuck in this subject area. It tells you where and gives you examples of where you might find different fungi on bryophytes and discusses them really, really nicely. So yeah, I'll talk a little bit more about some of this stuff in the examples coming up. Now, so here's just a snapshot of the biodiversity of these fungi. So I'll talk about fungi on liverworts first and then fungi on mosses. Just remember that liverworts are two-dimensional, generally speaking. They have their leaves in ranked rows, and this makes them a lot easier to screen for fungal infections than mosses, which you have to play around a lot more, dissecting to see things a lot of the time. So no talk on Bryophilous fungi would be complete without talking about epibryon, which are probably the most frequent fungi you'll find on liverworts especially, and on mosses too. There are really successful, hyperdiverse, probably, group of fungi, some of which are able to grow on multiple different plants, despite being very similar morphologically. You can see them here on almost every leaf, including some of the really young leaves of this liverwort, Plagiochyla. And if you look at them under a compound scope or a bit closer, you can see that they look like kind of black sea urchins with little spines coming out of them, again, sitting on the surface of the liverwort leaf. And here, coming out of it, is the sort of hymenium, the reproductive part of the fungus that's kind of spewed out the side of this little hole that came during the preparation of this. And you can see the sort of spores inside there. And this is a picture at about 400 times magnification, cropped. So it's even smaller than this looking under the scope. And you need oil immersion to really see those spores clearly. And this image is cropped as well. So really, oil immersion is really necessary to accurately see and measure these spores, the reproductive parts of these fungi, which are really useful for identifying them. Can be a bit of a hunt sometimes to see them effectively. Epibryon's interesting as well because you can use some chemical tests. If you treat preparations with potassium hydroxide and stain with Lugol's iodine, you can see a nice, really vivid blue reaction in a jelly that surrounds the reproductive structures of the fungus that come out of the fruit body. And this jelly is diagnostic of the genus Epibryon. So, and it's completely invisible in water preparation. So it's really nice color, and you can actually see these things. So if you stain them, you can often find the spores a lot more easily in these things than in water. I should also say these fruit bodies are desiccation tolerant. So if this plant dries out, these fruit bodies are able to dry out as well and survive at the same time. Many other species on liverworts have leaf perforating fruit bodies. So these two species in the genus Bryocentria, which grow on very common epiphytic liverworts, have this strategy basically where they grow underneath the leaf or underneath this ribbon-shaped part of the plant, and do most of their development there in this nice sheltered place away from sort of the desiccated, sort of dry environment at the surface of the plant. And then when they are larger, they actually rip through the plant tissue, as you can kind of see here. And very vaguely on here, you can see little spots kind of where the fruit body is torn through the tissue of the plant. And then they release their spores into the air or onto the plant, and they can blow and infect new plants. So really quite a cool strategy that these fungi use. And these fungi are probably very common, especially in South England. So here's a map, or here are two maps basically of, not quite, but almost the places I've been on field trips, because I'm pretty much the only person looking at these things other than an Irish record and a Welsh one. I think all of these records might be mine. So I'm pretty sure that this fungus is also present in other parts of England and Wales and Ireland as well. So, and this one certainly, if it's found in Scotland, up there on the Isle of Arran, and then down in South England. If any of you are looking in Britain, I'm on this really common liverwort, I'm sure you'll find this fungus no problem in other places. So anyone who does see it, please let me know. There's a lot of scope for finding new biological records all across Britain. This fungus, Bryocentria hypothalina, was only described as new to science in 2010. So yeah, there's a lot of quite new stuff coming out on these things. This is a parasite I really love, Periantria, which attacks the same liverwort, one of the same liverworts I just talked about, Frillania, very common again. And what Frillania does to reproduce is it has these leafy shoots, which are normal for the plant. And then they produce these female reproductive organs, which are these warty perianths, they're called, which I've arrowed in the pictures here. And inside these perianths, the sporangium of the liverwort develops, which is the sort of reproductive part of it. And when the sporangium is mature, it comes out of there and it forms these really nice flower structures when they dry out and release their spores. It really almost looks like a little flowering plant. And these spores flick away, these sporangia flick away spores into the air, and new plants can grow on new trees, whatever. But sometimes if you look at this plant in the wild, you can see whitish perianths with no sporophytes coming, or sporangia coming out of them. And if you look really closely, you can see that there are fruit bodies, just like those Bryocentrias I just showed you, that perforate through the perianth, and they can make a hole in it, as you can see here, and they shoot out their spores. And what's remarkable about this fungus, what it's doing, is it's exploiting the natural reproduction of the liverwort. So what happens in these species is that the leaves photosynthesize, and they provide nutrients to the developing offspring in this sporangium that's immature in this case, and it funnels all these nutrients into here. So this perianth with its, which is a sheath covering the sporangium, is protecting and nurturing the offspring in here. And what the fungus does is it grows inside, or gets in somehow, and hooks itself up to this nutrient stream and steals all the nutrients for itself, forming its own fruit bodies in the place of the liverworts one, which just ends up being a mummified husk. And then the fungus will go on and reproduce itself. So really a remarkable, super specific attack mechanism here by this fungus. And it completely does not harm at all the vegetative sort of part of the plant that it grows on. It's only these reproductive organs, which is just amazing. And I really just love this fungus. It's actually, I think it was my Twitter photo for a very long time. So moving on to some fungi on mosses. Now remember, mosses are a bit more 3D than liverworts, which makes them a bit more difficult to, or a bit more fiddly anyway, to look at for fungi. So you get some delicate ephemeral fungi which can come up on mosses. So you get a lot of sort of necrotrophic parasites that will attack and kill mosses, such as this Niacea nivea. And you can see here, this moss is really not having a good time. It's red, it's stunted, it's accumulated pigments, and its growth is not good. And when I collected this specimen, I didn't actually notice the fungus at all. I noticed that this was this weird red moss growing on the ground, and I'd never quite, it didn't quite match anything in my head. So I wanted to have a closer look, because this moss is usually green. So these fungi can really impact the way the moss looks and grows. And this fungus only comes up in the winter, in the wettest months of the year. It's if you were to dry it out after maybe just a day, it could be completely dead. They're not very good at surviving in warm conditions. These fungi are very good at surviving in warm and dry conditions. Bryostroma and acrospermum, two different fungi completely, that are able to grow on mosses that live, say, on trees in this case, which can dry out very quickly, very variable environment, and also on sort of chalky soil. And these fungal fruit bodies have thick shells around them, protecting the delicate spores on the inside. And when the moss dries out in dry conditions, the fungus is able to keep its structure without being sort of crushed by the effect of water leaving its cells, which is a really something that both plants and fungi really had to struggle to come up with solutions to when moving onto land, probably back in the day when they did that 500 million years ago. So you can see this kind of really nice co-evolution here, these club-shaped fruit bodies and these sort of big, thick, round ones. Now, many naturalists will be familiar with Polytrichum, which is a genus of mosses, the quite big mosses that grow out in woodland and also in bogs. These are really excellent hosts for ascomycetes. So Polytrichum are quite different to the other bryophytes I've talked about, because they have multi-layered leaves. They're not just a single cell layer, they have these files of cells. And if you cut a section of the leaf through the middle, you can actually see that these files of cells called lamellae are photosynthetic. And then below that, you have kind of storage and structural layers of the moss. And fungi absolutely love these lamellae, these photosynthetic regions of the moss, and they'll form their mycelium in there, and they'll form fruit bodies in these gaps. And the more observant of you may have noticed that there are fungi coming through the gaps of this leaf that I showed here. And yeah, fungi love it. There are dozens of species that have all convergently evolved to grow in this, the leaves of these Polytrichales. So amazing. I mean, almost every Polytrichum you find in a forest will have this fungus on it, I guarantee you. Another fungus which is super interesting is Lawsonia. So I've told you about a fungus on liverworts that kills the female plants and the reproductive offspring. Well, this one does the opposite. It kills just male plants. So here's what male plants of Polytrichum normally look like. They have these brightly colored or green male sort of splash cup heads. And this fungus attacks these heads, kills them, and turns them black. And very specifically, you can see all these healthy green plants and all these blackened heads here. And if you look closely, you can see that the black is actually caused by lots of little black fruit bodies really aggregated in here that look like little cannons with little cannon heads aimed out. And these will shoot spores out and infect more plants. And again, pretty much specific to male plants. You do get a bit of variation in that. And there are about 4 or 5 species, a few undescribed ones, of these parasites. They're very common, actually, if you look for them. But many people just ignore these, thinking that they're just dead heads. So there are very few records of these things in the British Isles. And one cannot talk about Bryophilous ascomycetes without mentioning the Bryophilous or bryoparasitic pyronemataceae, so species like Lamprospora and Octospora. And these are super common, and some mycologists will already be familiar with them. And they're basically orange discs that grow next to mosses. They're not the most glamorous fungi, none of these things particularly are, at least macroscopically, but microscopically, they're amazing. The spores are just so diverse, and they're very host specific. So you can actually, as a mycologist, you can learn your mosses partly by looking at these fungi, because the spores are very different depending on the host moss. So there are hundreds of species of these. Some are specific to a single moss or to some closely related mosses, and that host specificity is often mirrored by differences in the spore morphology and the ornament of the spores. So some of them are smooth, like this one. Others have curved ridges or look like asteroids, or these bit of ridges and sort of puncta on the surface. This one has spines. So really just amazing. And as a mycologist, I'd really recommend you, if you want to start off with these things, is to go into these. In the last 6 years, we've had a sort of 50% increase in the number of species of Bryophilous fungi found in the British Isles, plus many undescribed or cryptic species. So I'm writing a manuscript trying to describe the species currently known in Britain, but because we keep finding more all the time, it's quite a project that just keeps getting bigger and bigger. But I'm hoping to draw the line in that soon and put a resource out into the community that might help people get involved, because they're not really any guidebooks or anything for these things. All of it's really in the primary literature. So I'd like to kind of collate those data and present something useful to the community. So finally, I'm going to talk about what we can learn from studying these things, the interaction side, not just going out and finding a cool species, which is really fun, but what else can we learn from them? So the key question for me is how plants and fungi have co-evolved on land. And many people now know that these fungi called arbuscular mycorrhizal fungi, or glomeromycota, are really important in plant health, really. So many trees will interact with these fungi, and the fungus will give the tree nitrogen and phosphorus, and the tree will give the fungus carbon. And if you take the fungus away, the tree will die. So you have these symbiotic interactions between plants and fungi. And all major plant lineages really have these interactions. So they were probably present when plants originally colonized land. And you can see them in liverworts, and people have known about them for 100 years. But ascomycetes also interact with bryophytes and form mycorrhizal associations with plants that might involve nutrient transfers. And some of these, called the ericoid mycorrhizae, are particularly interesting because you have a fungus, for example, that can interact with both liverworts and ericoid plants at the same time. The same fungus can interact with a flowering plant and a bryophyte, possibly connecting the two in the soil. So, wow. I mean, isn't that just a really strange thing? Wouldn't it be great to know more about these? Something I find really cool is the fact that there seem to be lichenized bryophytes. So I mentioned earlier that lichens are an association between a fungus and an alga or a cyanobacterium, but there are many instances where lichen fungi have lost the algal component and have sort of lichenized, if you will, bryophyte cells. This has happened in the genus Protothalonella, where some species grow as lichens on the soil, others grow as lichens on moss leaves like Protothalonella petri. And then the one I haven't seen, Protothalonella polytrichi, seems to have lost the algal part of the lichen and actually grows, its mycelium actually interacts with the moss cells instead of an alga, which is remarkable. I mean, would you call this thing a lichen then, or would you call it a moss? It really stretches the definition of what we classically would call a lichen. And the same thing has happened in many other groups like Catenaria, where this species as well seems to have lost the algal component of the lichen relationship and is sort of lichenized a liverwort. And finally, I'm interested in how fungi influence bryophyte biology. So here is a fungus called Naecia youngermaniae, and it forms these nice blue apothecia. And a study found that this fungus actually manages to influence and manipulate its host's development. So normally a host would produce a reproductive structure like this. And when it's infected by the fungus, this is the fungal fruit body, it makes the structure completely change its morphology. And the fungus basically causes the part of the plant it sort of feeds on to over-proliferate, to make more of it, so that the fungus can make more of itself and reproduce. And a similar thing goes on with this Octospora, which is parasitic on the rooting system of a moss, and it makes this rooting system over-proliferate and form these gall-like structures, which here are wrapped around by fungal tissue. And so the fungus is siphoning nutrients away from the moss by manipulating its development. And this stuff is something I find very interesting and worth exploring more in the future. So really today I want to end off with a plea to people. If you'd like to contribute towards uncovering the biodiversity of Bryophilous ascomycetes, I guess in Britain, but also beyond that, please get in touch. There are several, we're already, getting quite a lot of data on this. I've got a sort of herbarium now with about 400 to 500 specimens collected by me and also dozens of other naturalists across the world, actually, which is great. Especially Jamie Warren on Twitter, just a shout out to him because he has collected a lot of really interesting species, including some new for Britain. I'm trying to encourage further interest both on Twitter and also via my website, which you could go on to if you like. It has species found in Britain and Ireland so far. And I also want to explore some of the understudied places that haven't really been looked at, habitats for Bryophilous ascomycetes, such as oceanic regions of Britain and Ireland, the west coast of Scotland and Wales in particular. And at the moment, we're kind of starting off I got a little bit of funding from the Bristol Naturalist Society very kindly to start in my spare time sequencing some of these fungi so we can start to understand where they belong in the greater scheme of fungal sort of systematics. So yeah, with that, I'd like to thank you all for listening. I really appreciate you coming along. I'd like to acknowledge all of these people, Colin Pope, especially Peter Derbler, Paul Cannon, Marcel Vega and Lucas for our recent field trips in the Alps, John Norton, and again, all the naturalists who've sent me specimens and accompanied me on field work. It's too many to name, but really thank you all. Universities of Bristol and Oxford, my PhD supervisor, Andy Bailey, Sebastian Shornack, the Bristol bunch, and all these societies for their support over the years. If you want to contact me, please do it via Twitter, or the contact form on my website leads to my personal email address, which I'd rather not put on because this talk's recorded. And if you have any questions, now is the time. Thank you again.
Thank you so much, George. What an amazing journey through your side interest in this subject. Amazing. I mean, goodness me, what a body of work. Firstly, I was just going to repeat that, yes, this is a recorded presentation. So George has shared with us quite a number of references and suggested social media sites and things like that. So you'll be able to, if you haven't noted those down, you'll be able to check back on the recording, and that will be found on the BMS YouTube channel in a couple of days, hopefully. So we have got some questions in the chat, and I'll go back to the beginning of those and just ask you, I'll read them out, George, so everyone can hear if they can't see those. So first of all, from Flavia, are these, two parts of this question, are these fungi culturable? How and in which media?
So some of them, yes, others not. And it really depends. I think some of the ericoid mycorrhizal fungi might be culturable in sort of some standard media, maybe PDA. But really, I mean, I haven't really worked much on that aspect of stuff, so I can't really comment on it at this point. But there are papers on it, so if you want to look, I'd suggest going into some of the references I've cited.
Super, thank you. Flavio also asks, the symbionts, are they obligate?
Some of them are and some of them aren't. Like that, again, Rhizoscyphus ericae or Hyaloscypha ericae, which is this ericoid mycorrhizal thing, can seemingly interact with both bryophytes and flowering plants, but others are specific to bryophytes and only bryophytes.
Okay. Next question here is from Sim. Now, excuse me if I stumble over this. It's a tricky one. Are any of the Bryophilous ascomycetes also licaniculous, yes? Or are Bryophilous and licaniculous fungi different species?
So that's a really interesting question. And the answer at the moment is for the vast majority of them, they are separate. I think there are a couple of exceptions where some really generalist fungi can attack a lot of different stuff. Mm-hmm. But bryophytes are plants, and lichen, well, the things that lichens generally are alga and fungus. So a lot of the fungi that have evolved to attack lichens are often actually fungi that can also attack other fungi. So yeah, there's not a huge amount of overlap.
Okay. A question now, you shared quite a few images at different scales here, and there was a question came in a little while ago asking, can they be seen with a hand lens? And I'm not sure exactly which images we were looking at the time, but what generally is the scale of some of these fungi?
Generally very small. Again, about many of the ones I study are between maybe 100 or even less than that and 250 micrometers in diameter, which is up to 1/4 of a millimeter. So you will need most of them a hand lens to see them, but sometimes that's not enough. This one here in the picture, you can see with the naked eye. So it really depends, but yeah, they are small mostly.
Okay. There's a question here from someone who's been finding a lot of microplastics embedded in lichen samples and wonders if the fungi are using microplastics to infect the bryophytes. So perhaps if the spores could be stuck to those microplastics. I
guess it's possible, but it's never been investigated empirically. So yeah, it's definitely possible.
Okay, thanks. A lot of comments saying thank you very much for your presentation. I'm just whizzing down now to see. Here's another question from Danny. Danny, how much work on these fungi has been conducted in the tropics, particularly in the neotropics?
A bit, but not that much. It's mainly work by Peter Derbler, a little bit of field work, some herbarium work. I think it's pretty much exclusively by him, actually. So I expect if you were to go to these places, you will find a huge number of undescribed species.
Mm-hmm. Mm-hmm. Cameron would like to know when your manuscript for identification is coming out. Cameron says he's made many attempts to identify Bryophilous fungi. However, the concise literature is pretty limited, and accessing research papers is not always so easy.
No, it isn't. Yeah, that's a work in progress. It takes a long time because it depends on, it's already almost the length of a book. Yeah. So do I want it to be a book or do I want it to be a paper? Do I want it to be freely accessible to people or do they have to pay for it? How detailed? A lot of questions. I need to actually talk to people about it at some point soon. If anyone here does want to talk about that, do let me know. But my website is trying to be a freely available source of information currently that anyone can go onto and look. And it's got host indices as well. So they're not always fully up to date, but it's got everything you'll find in Britain so far. So do have a look at that if you are keen.
Okay. Ryan Douglas says, given that around 22.5% of bryophytes are considered threatened in Europe, what do you think about potentially red data list assessing host-specific Bryophilous fungi as threatened?
Yeah, I think it would be really good because for example, there is a threatened moss on the Isle of Wight and it grows in two, I think, places. And there is a fungus that is specific to that moss that grows right next to it and only on that moss. So that fungus should automatically get the host, the conservation status of its host, but no one does that.
Mm-hmm. Okay.
It might be a bit redundant if the host already has protection, but I don't know. Yeah, it's a good question.
A question here from Christian. Christian says, I know this is a substantial step away from ascomycetes, but it seems that there's a growing awareness of hygrocybe, this is testing my ability to read out these Latin names now, Entolemataceae, Geoglossum clavaria forming close association with bryophytes. Is that the case? Mm-hmm.
I've heard a bit of that, like with Geoglossum as well. But again, I guess I study the small stuff at this, even smaller stuff than that. Yeah, I mean, it's really interesting that's there are clearly actually a lot of basidiomycetes that are Bryophilous as well that we are finding a bit more about. So it'll be interesting to study that. I mean, I've had my hands full with the ascos. So yeah, anyone else is welcome to take on the basidios.
Okay, a couple more questions, I think. There's one here from Joyce. When a fungi parasitizes a moss and uses it to develop a fruiting body, have the spores ever been collected and analyzed for DNA to see if some of the moss's DNA is in the fungal spores?
I don't know that. So I mean, people analyze, have done a bit of sequencing of these things, but whole genome sequencing, I would say not really. It's likely that there's been gene transfer between, you know, the mosses getting fungal DNA and the fungi getting moss DNA. It's very possible, but there haven't been studies confirming this as far as I know.
Okay. A question here about how to send samples and to you. So from Anthony, in terms of preserving these to send to you or other enthusiasts, are they better sent damp to keep the moss fresh or dried to stop any decay?
It depends on how quickly they get sent, partly, and whether it's worth the risk of sending it hydrated, because that the samples could get moldy, but also, as is the case with these octosporas, they often get eaten by invertebrates that are living on the moss that will munch these fungi, and you'll get the sample in the post and there'll be nothing left. So drying it out obviously is bad in some ways because it reduces the amount of morphological characters that are useful. Some stuff will just deteriorate in dry material, but it's the safest route. So sometimes a hybrid approach of a bit of both is maybe best.
Yeah. Okay. Yeah.
Yeah.
Same person, Anthony, second question. Are there any easy keys for beginners to identify mosses and liverworts with minimal jargon, diagrams, what to look for, that kind of thing?
I would say go to the British Bryological Society's website. They have a lot of tools and tips for beginners. They also have a field guide which tries to be, which I think lists common bryophytes in different habitats, which is very helpful as well. Okay. Yeah.
Okay, just one more question then before we finish for this evening. Interesting question. As permafrost melts and some ancient mosses come back to life, do the Bryophilous fungi come back to life as well?
We don't, well, that's interesting. We don't have evidence of that I know about, but there are mosses, and there's a fungus I showed earlier, that live in snow beds. So every year they get frozen over and completely, and they're in this battle and they're just frozen together. And then it thaws out for a few months in the summer or whatever. This is, say, the top of the Cairngorms in Scotland, or maybe in the Alps as well. And during the summer, everything comes to life and they're whacking at each other, killing each other, trying to grow and having this battle. And then the snow comes over and freezes everything again. So I imagine that in a larger time scale, the same thing could happen with permafrost potentially. Yeah.
Okay. Thank you very much. I think we'll draw the evening to a close there. Thank you again, George, for giving us such a fascinating talk on this, what is at the moment a niche area, but I think there are plenty of people here tonight who will be encouraged to pursue this further. It's been amazing. Thank you.
Oh, thank you. I hope so.